Alright, mitosis is the process that cells use to divide, and it's important that cells be able to divide so that you can grow new cells. And in mitosis, you start out with one parent cell, and it splits and becomes two identical daughter cells, which means they have the same amount of DNA and the same type of DNA. And mitosis is followed by a process called cytokinesis, and this is the process where the cytoplasm of the two cells actually divides. So at the end of mitosis, they're not quite split yet, but after cytokinesis, then both cells are completely individual. The first step of mitosis is prophase, and prophase is the step where the genetic material that's in the cell condenses, and it's going to form what looks like little X's. And so the genetic material is usually kind of stringy. It looks sort of like a ball of yarn. But during prophase, the genetic material is going to condense, and the nuclear envelope breaks down so that genetic material, those chromosomes, are going to be able to move around the cell because the nucleus has dissolved. After prophase, we have metaphase. In metaphase, you have spindle fibers starting to form. Spindle fibers are these spider-like things on the edge of the cell. And the spindle fibers are going to attach to the centromere of the chromosome. The centromere is right in the middle of the chromosome. And they're going to push the chromosomes to the middle of the cell. And so they're going to make a line right down the middle. So in metaphase, you can always think of the M in metaphase for middle because the chromosomes line up in the middle of the cell. After metaphase, we have anaphase. And in anaphase, those spindle fibers that are attached to the centromeres of the chromosomes are going to start to pull apart, and they're going to break the chromosome into two sister chromatids. So when it's together in an X, we call it a chromosome, and when they separate, each of those halves of the chromosome is called a chromatid. And those chromatids are going to separate and move to opposite sides of the cell. Anaphase starts with an A, so you can remember the word away with anaphase because the chromatids will move away from each other. After anaphase, we have telophase. And telophase is sort of the opposite of prophase because in prophase, the nuclear envelope breaks down, while in telophase, a nuclear envelope forms. And there's going to be two separate nucleuses that start to form, one around one set of the chromosomes and the other around the other set of chromosomes. And instead of being in a nice compact X shape, the chromosomes are going to start to break down into chromatin, which is sort of stringy DNA, sort of like a ball of yarn. And so this is the way that the DNA is going to be most of the time in the cell, except for when the cell is dividing, which is when it forms chromosomes. And remember, after telophase, we have cytokinesis, which is actually where the cytoplasm will completely divide. So we see the cell membrane starting to form, but there's still some connection between the two cells. They won't become two individual cells until after cytokinesis. And cytokinesis is not considered to be a part of mitosis, it's just a process that comes right after mitosis. We're going to go through the cell cycle, and in the cell cycle you have several different stages. We're going to begin with the G1 stage, and in the G1 stage we have cell growth beginning. So this is the life cycle of a cell, so it has to start by growing, and the cell's also going to get ready to copy its DNA. So in class we did an activity where we had pieces of paper to represent chromosomes. We put those pieces of paper into a bag, and that represented our cell. And then to represent the cell growing, we're going to add some beads to the bag until the 5 grams. And so our parent cell that's completed its cell growth, it weighs, well, in the picture more than 5 grams, but in class we made them weigh 5 grams. After this part of the G1 phase, we're going to enter our first checkpoint, and our first checkpoint is called Ras cyclin. Ras cyclin is the checkpoint where the cell makes sure that it's big enough, that it's gained enough mass to go into the next phase of the cell cycle. And so your cells actually have these points in place to make sure that nothing goes wrong in the cell cycle. And so I came around and checked to make sure that all of the cells had grown to the right mass so that they could continue on. And we would move on to the next stage of G1, and we come to another checkpoint. In this checkpoint, it's called P53. This is where the cell is going to make sure that the DNA of the cell is not damaged. And so I came around and checked to make sure that none of the DNA was bent or torn, and then we were ready to move on to the next part of the cell cycle, which is the S phase. Now notice that G1, S, and G2 are all a part of interphase. Interphase is a very long phase in the cell cycle, and it consists of these three parts. So now we're on the second one of those. That's the S phase. In the S phase, the cell copies and synthesizes new DNA. So this is where your copies of DNA are made. And so we took the pieces of paper out of the bag and made copies of them. So we now had 8 pieces of paper instead of 4. And we put them in an X shape because your chromosomes, after they've copied themselves, will form an X-shaped bundle of DNA. And so we would take those 4 and put them into X shapes like this. So now we have our 4 copied. Once the cell has copied the DNA, then it's ready for its next checkpoint, which is called ATM nibrin. And at this checkpoint, the cell makes sure that the DNA was copied without any mistakes. And so I came around and made sure that all of the DNA that was copied was the same size and length and color as the DNA that they started out with. Then we're ready to move on to the G2 phase of the cell cycle. And in this phase, the cell gets ready to divide. So we took the spindle fibers out of the materials bag and prepared for mitosis. This is what the spindle fibers look like. And then we're going to move into the cell division portion of the cell cycle. So we've now completed interphase. We're going to begin cell division. Another name for the cell division part of the cell cycle is the M phase, and that M comes from mitosis. And so the cell division, or the M phase, includes mitosis and cytokinesis. So we're going to go through mitosis first. Mitosis starts with prophase. In prophase, the chromosomes condense. So we used a pencil to curl the chromosomes so that they got smaller. And then we moved into metaphase, where the chromosomes line up in the middle of the cell. So we lined up the chromosomes in a line on the desk. Then we moved into anaphase. In anaphase, we attached the spindle fibers to the chromosomes. We attached one side of the spindle fibers to the top chromosome and the other side of the spindle fibers to the bottom of the chromosomes. And then we were ready for our next checkpoint, which is called MAD1. At this checkpoint, the cell makes sure that the spindle fibers were attached correctly, and this ensures that the chromosomes are divided evenly and that you don't get any that don't make it to the new cell or anything like that. And so I came around and made sure that all of the spindle fibers were attached correctly. And then we completed anaphase by pulling the chromatids away from each other so that they ended up on opposite sides of the cell. And then we moved into telophase, and in telophase we remove the spindle fibers and we leave the chromosomes in 2 piles. And the cell hasn't completely divided yet, so we still have some cytoplasm that's connected there, but we're going to start to have a nuclear envelope form around the chromosomes. Then we get to move into cytokinesis, and in cytokinesis we have a full division of the 2 cells, so we get 2 completely new cells and the cytoplasm divides. And we symbolize this by putting those chromosomes into a baggie, so we have 2 new cells. And you can notice that the chromosomes in the daughter cells are going to be identical to the chromosomes that you had at the beginning in the parent cell. And so that's what you get in mitosis. You get 2 daughter cells that are identical to the parent cell. Now, when there's anything that goes wrong in the cell cycle, especially at the checkpoints in the cell cycle, you can end up with uneven numbers of chromosomes in some of the daughter cell baggies. You might end up with chromosomes that are damaged or that weren't copied correctly, or maybe too many or too few chromosomes in each bag. And this is where cancer cells come from. This is the result of mistakes in the cell cycle. And so when those checkpoints don't function correctly, you can end up with cancer. And so that's why those checkpoints are so important, because they prevent cancer cells from forming. They keep the cell cycle running correctly.